Right, it's now 11. So I think we'll get started. Hi, everyone. Welcome to our Ask the Experts webinar on navigating antibody characterization with LCMS. Today we have two speakers, Dr. Jinrui again and Anthony Stajduhar. So just a little background on them. Dr. Gan has earned her PhD in analytical chemistry, specializing in the development of analytical methods to enhance sensitivity, selectivity, and efficiency in proteomics using mass spec. She has over 13 years of expertise, and she has applied mass spec techniques to advance protein and antibody characterization. Anthony is the Director of International Business Development here at Rapid Novor. He collaborates closely with researchers in antibody characterization, discovery, and diagnostic development, gaining a deep understanding of their long-term goals and specific requirements. He has a solid background in engineering, and Anthony is dedicated to solving challenging technical biomedical problems while enriching the lives of humans around the world. If you have any questions during this webinar, Anthony and Jinrui both have some slides, but you can use the Q&A function in the bar below and submit all your questions, and then we will all address them towards the end. And without further ado, I will let Anthony take it away.
Great. Thanks a lot, Kenya. Appreciate your help. Hello, everyone. Thanks for coming. Appreciate your interest and hearing a little more about us. We thought it'd be a good idea to just do a little bit of introductions into Rapid Novor for those of you that haven't heard of us before, or maybe new to our company. Rapid Novor has been around since 2015. We really specialize specifically in mass spectrometry. In particular, we've always specialized in the use of mass spectrometry towards sequencing, protein sequencing, canonically for like an antibody, but we also work on a lot of different proteins as well. And over the years, we've been able to grow to a fairly decent sized team here in Kitchener, Ontario. We've got over 60 employees, 10 different teams, over 20,000 square feet of lab space, and over 15 mass specs in the lab. But although we specialize in protein sequencing, and we've been able to do that for many clients worldwide, with over 9,000 different monoclonal antibody, polyclonal antibody sequencing projects, with most of the major pharmaceutical companies, many smaller biotechs, many academic institutions. We've been able to sequence polyclonal antibodies as well, wherein we find the most abundant monoclonal antibodies within a polyclonal antibody mixture. And we've been consistently innovating now over the past several years with over 400 different clients across the world. With all that being said, we've been able to extend some of the services that we've offering to our clients, away from just exclusively like protein sequencing based services and now into more routine mass spectrometry workflows, antibody characterization type analyses, such as what we're going to be talking about today, peptide mapping, disulfide bond analysis, PTM analysis, glycosylation work, as well as sequence variants. But in addition to these workflows, we've also been able to roll out an HDX mass spectrometry epitope mapping service, as wherein we can discover the amino acids responsible for binding between two molecules broadly. Canonically, it's like a protein-protein interaction, antibody-antigen kind of situation. But of course, like other more complicated constructs are also possible. Small molecule to a protein of interest, or a DNA, strand of DNA even to like a protein of interest. We're always happy to discuss and consult on a prospective project, as well as like SPR services and some of our more recent cloud-based approaches towards mass spectrometry software. But today's focus, as I said, was more on antibody characterization. And so sometimes it's pertinent to kind of just explain why we might want to be interested in characterizing proteins and antibodies more generally. So certainly, I think we could all agree that biologics are, by and large, a little bit more complex, less stable, not as easily identifiable as a chemically defined, a small molecule or chemical of interest, and thus they need to be monitored more closely, especially over time, as antibodies can degrade and change and mutate essentially with post-translational modifications, just by virtue of being in a vial. And so thus, antibody characterization is really quite critical in both downstream regulatory activities and ensuring that the molecule that you're putting on the market or that you're putting into the clinic is the same when it's leaving the factory as it is when it's entering, a human body. But even earlier than that, it's also quite pertinent in terms of the early stage discovery work as well. We're try to make the case here of, we understand there's a wide audience in this group. So some of you folks may be from the downstream side, more on the CMC side, and some of you folks may be on the antibody engineering early stage discovery side. So we're trying to address, the needs of like both of these, both of your communities like in the webinar as well. So indeed, like there is the requirement obviously for ensuring regulatory submissions and ensuring like product robustness and um, activity and safety long term. But indeed, these considerations are just as valid at the early stage. Removing those issues downstream could be just as valuable as needing to address them when they are downstream, right? So what we are trying to avoid are situations like this, which is a pretty well-known case. This case with Ipsen, wherein they actually had to deal with a phase 3 clinical trial hold on their treatment for hemophilia B, and in particular is due to the unexpected presence of host cell proteins from a CHO expression cell line. And similarly, in a prospective COVID-19 vaccine, there were similarly un unaccounted for molecules that are present in that particular sample as it had been manufactured. And so really we're trying to make the argument that like double checking your molecules is obviously quite useful before they go into the clinic, but another set of eyes, a complementary viewpoint on characterization services and their workflows, like maybe of great utility, not only in the downstream, but also, as I said, in the upstream. And from a regulatory perspective, we can understand that there are essentially, two different colliding um, aspects to the stability of your molecule of interest, right? Because on the one side, we're always concerned about the efficacy of the molecule. Certainly, any degradance of the let's say an antibody of interest could potentially lead to the loss of efficacy of that molecule. But similarly, the increase in degradants could also lead to lack of a loss of safety in that molecule as well, as the aggregates themselves and the degradants themselves could actually be potentially immunogenic and cause issues downstream for the patient receiving that therapy. And so we acknowledge that there are differences between impurities and degradants. Impurity would be something like, what I just referenced earlier, like the host cell protein, some other material that you don't expect to observe in the sample of interest, whereas degradant is really something that is changing the molecule of interest, such as a post-translational modification, which we will be able to address quite I think Jinrui will be able to address this quite extensively in her talk about things such as deamidation, oxidation, proteolysis, and these kinds of concerns and issues that you can observe in the molecule of interest. And so there are many different process-related impurities that we have to watch out for, residuals from cell culture, from the cell substrate, issues from purification, seeing other proteases or nucleases. And it's interesting because sometimes these don't just show up in the final product, or sorry, the produced product, but rather after the formulation. We've been working in this industry for quite some years, and it's funny to hear some stories that sometimes the product looked great coming out of the hands of the cell culture team and the manufacturing side, but then when they put it in the formulation, things changed a little bit because sometimes buffers and formulations can alter the substance itself as well. So there are always things that we need to look out for, and it takes a trained eye and a skilled eye, which is why we're really happy to have Jinrui on the team to help with these kinds of requests. And of course, these are certainly relevant with respect to the antibody characterization from a regulatory perspective. For those of you from the CMC side, I'm sure you know that you need to submit these kinds of documentations, and whenever we're submitting paperwork or trying to justify the reason why we did a particular assay, it's always important to see where it is written. And indeed, characterization is written here. These are the actual guidelines from the ICH, and they are quite keen on demonstrating that the antibodies or the proteins of interest were characterized and run in a fashion which is like repeatable, robust, and ultimately valid in the eyes of regulatory bodies. And so while we are at Rapid Nova, we're not a CGMP or a certified laboratory to that extent, we are always happy to make the methods and materials that we use in our protocols available and to our clients at the end of a project. And this is ultimately, I think, in the interest of anyone who's trying to submit something to the regulatory bodies. They may not always need CGMP grade analytical investigations, but nonetheless, they certainly encourage the use of characterization, services, analytical workflows towards the goal of characterizing, a prospective clinical molecule, even if it's the early stage, like sharing that information, sharing your due diligence, sharing the fact that you've been able to analyze these molecules, even from the very early stage, is quite useful from their perspective in demonstrating that, your goals are aligned with ultimately the FDA's and other regulatory bodies like the EMA's in the long term in terms of like truly characterizing robustly that these molecules you're producing are, what we think that they are and doing what they should be doing. And so there are many ways in which one could characterize antibodies, of course, and a simple example would be to be using glycans on an antibody of interest and answering a question like, is the recombinant antibody that I'm working with recapitulating the same glycan motif as the original antibody in a hybridoma? This is like an early stage antibody discovery question. A lot of people are still using hybridomas. Of course, B-cell antibody discovery platforms have become much more popular, but the same question still kind of holds. Is like this recombinant antibody acting the same as the antibody that I, pulled out of a transgenic animal, pulled out of a human sample set? Are we really observing the same molecule? Because these are questions that you can't necessarily get out of just doing like an NGS sequencing or even because the post-translational modifications will not be encoded in the nucleotides, and thus they may not be obvious. And it's certainly been the case that we've observed that with some of our clients who've come to us seeking, for example, a protein sequencing project for a monoclonal antibody. They send us the original that came from a hybridoma, for example, and there may be an additional glycosylation site located in a CDR region or variable region, or just like an O-link glycan that's unexpectedly there. Whereas the recombinant isn't producing that. They're losing the binding activity. They're losing the efficacy essentially of their molecule of interest. So both on the later stage in the regulatory environment, but also in the early stage, sometimes having an extra set of eyes, using a mass spec to answer these kinds of questions can be of really great utility in order to ensure that you're really recapitulating what you were initially observing in your antibody discovery and engineering workflows. But of course, that's just one aspect of antibody characterization. There are so many applications, so many tools. And although we've whittled it down to just a handful here, I think the really pertinent point to this is that it takes an expert to really use those tools correctly. Many people can wield a hammer, but how many people can build a house? I think is like an analogy that I could use here. And I'll let Jinrui jump in a little bit more detail about how we may build a house for you. Think you're on mute, Jinrui? Okay.
Yeah, it's hard to navigate the button on the screen. So yeah, hi everybody. Thank you, Anthony, for the background introduction. So as you already can know that it's pretty, like I would say, quite important to do some type of characterization as early as possible to avoid the later stage of, redo of everything, and especially with the engineering and everything. So so here you can all see the slides, right? So here is the whole panel of the therapeutic antibody characterization that we normally do during early stage, and also CMC together with the HDX that we also offer here. So this kind of all these services kind of together it covers most of the characterization you will need for during the lifetime of the antibody characterization. And so based on the characterization, so I divided the whole characterization into 4 categories. So potency analysis that normally is done by SPR that we do offer here. And then the purity analysis mostly we care about the aggregates, fragments, and charge variance, and also the process-related impurities such as HCNA, HCP, and protein A. So here, HCP is normally done by ELISA, and as you have already heard from Anthony, that HCP is really important, and also there are currently more awareness of using LC-MS, more application of using LC-MS to do HCP analysis. And that's something that we are also working on, but not yet live at the moment. And then the third category is a primary structure analysis, as you can see from here. And those are majorly done by LC-MS. You will be looking at the molecular weight, protein sequence, and PTMS, glycosylation, and then also the amino acid composition. And then the last, not least, is the high order structure. And one of them will also be done by LC-MS, which is a disulfide bond analysis. And you will be also sometimes looking at the free thiol analysis. And then the CD will provide a secondary and tertiary structure analysis. So DSC and DLS will provide some thermal stability and aggregation temperature analysis. So not all of this will be done. So all those analyses will be done. You would choose your analysis depends on the stage of your development and then to solve the specific questions you have. So in this talk, we will focus more on the LC-MS based, and then for we will talk about the following 5 different kinds of analysis: intact, disulfide, PTMS, chemical degradation, glycan, and the sequence variants. So so intact mass, we talk about intact mass is really, I would say it's pretty easy concept-wise. So you would use LC-MS. The LC can be a reverse phase, can be SEC, can be ion exchange. Anything that you can use to separate your protein of interest and the compatible with MS can be used for the molecular weight analysis. And here is a typical chromatography you will see. And then with the separation of your protein of interest, in this specific one is a heavy chain, light chain. And then you will get, for each of the one, if you either average them or analyze them separately, you will get a very good charge envelopes. And here, if you see, it's really like dense and it's like complex because this it has both heavy chain, light chain, and also have a different glycosylation pattern inside. So the whole spectrum is really busy. And after you deconvolute it, this is something you will be expecting to see with in the intact level. You'll be seeing that if the protein is glycosylated, you'll be able to see different kind of glycosylation pattern. So this is just an example of something we normally do for IND filing with the molecular weight analysis that we actually try to divide into 4 different 4 types of intact mass, which is like intact as it is. Here you get the result, and then if you know the theoretical molecular weight, you will be able to judge by the mass shift, you will be able to judge the major glycan form and what are they and what are the relative composition of them. And then by using the deglycosylation step, you will be able to see the native protein without the glycan. And then in the reduced level, you will be able to see the light chain, heavy chain separately. And then in a better, because now the mass is reduced, so you'll be able to see more in detail if some other glycosylation might come up in this level of analysis. And then in reduced deglycosylated, you will be able to see a deglycosylated heavy chain. Another level of analysis is middle down or middle up. So what we do is it's the middle up. So what we do is we try to, because the whole antibody is 150K and it's pretty big. So the solution, the arrow is mass arrow normally is bigger in that region, and so mass range. So here, what we were trying to do is try to cleave and reduce the whole antibody into 3 different subunits, FG, LC, and FC. Those each of them is 25 kD. And in this mass range, you will be able to get a much more accurate and much higher resolution, resolved spectrum, which give us more mass accuracy. Of course, this doesn't have to be in this specific order or this specific enzyme used. It really depends on what kind of questions you will have, what exactly you are trying to achieve for the analysis. And then the results of subunit will be subject to the LC-MS analysis. Here is just one example of how the subunit analysis will look like. So this is like two different antibodies. Then you see they are well resolved of the three units and also between them. And then the second section is example of how the zoom-in version of the FD region to look at the glycosylation pattern. As you see from the subunit level compared to intact, you can see much more in detail of the glycan form and also be able to see some really low abundance species that you would not be able to see in intact form, such as the lysine untruncated version with the G0F glycan modification. So those are just some tools that in the intact or subunit level to help us to understand to help us solve whatever questions you have. So the technique can be used to understand the DAR value, the drug antibody ratio value of the ADC drug. For example, this is a subunit analysis of the one ADC. And with the in this subunit level, you can see the roughly distribution of your drug payload. So you can see the light chain has the native one and one payload version. FD chain has three different kind of conjugation, different level of conjugation. And by calculating the allele peak area, we can get a relative quantification and then obtain the average DAR value of 2.9 for this specific ADC. And another application for those analyses is nowadays for bispecific antibody mispairing. This is coming from taking off from a reference paper. As you can see here, the corrected paired antibody is supposed to have two different Fab regions. But there is a chance of the LLC swap mispair species existing in the sample. And if you want to identify them or even quantify them, so you would cut them into subunit, in this case, in Fab and FC. So if your sample has the mispaired version and also the native one, you will be able to see three different mass distribution. One is the mispaired one coming out in one peak, and then the corrected pair one will have both of the Fab species existing in the spectrum. So, so those are the intact one. And then the rest of the analysis we do are majorly based on the peptide mapping workflow, which is with antibody, we do denaturation, reduction, alkylation, and digestion, and then obtain the digest, then subject to LC-MS with different kind of fragmentation, be that as ETD, HCD, and EDHCD, and obtain the data. And then this data will be submitted to a specialized software for data analysis. And here we use the PMI BIOS software for the data analysis. So one of the most common and also very important aspect of the characterization is the PTMS analysis and also chemical degradation. And so this is just a simple illustration of some typical kind of a PTMS or chemical degradation you will be expecting for antibody drug. As you can see here, N-terminal cyclization is really common. And then we also have methionine and tryptophan oxidation and asparagine deamidation, and also, of course, also the isomerization of the aspartic acid. Also glycation and lysine truncation, all those kind of typical PTMS will be analyzed during your typical antibody PTM analysis workflow. And this is just an example of how the data will look like. So this is like N-terminal cyclization. Since with this kind of a modification, you normally would expect the retention type shift of your wild type and the mutated of the modified version. So here the MS1 chromatography checked out. I did not show we also look at the isotope pattern. So theoretically in this kind of modification, you wouldn't expect the huge isotope pattern change. So the isotope pattern for the wild type and the modified one will also has to match really well. And then we also look at the MS2. It needs to have a good coverage and fragmentation. So when all of those three checked, we consider that as a true positive. So this is just an example of the C-terminal lysine, which is a similar case, just with the lysine truncation. You will be looking at the same criteria. So with oxidation, so the C-terminal, with the, let's say, C-terminal lysine, there's less chance of, we don't see much of the artifacts from the sample prep or the protein from the mass spec perspective. But for N-terminal cyclization, we sometimes also see artifacts from the really acid digestion version of it, and fraction of it, and also some cyclization will happen during the MS analysis. So that's something that you need to pay attention while analyzing the data. Another one important thing we see PTMS is oxidation. And as you can see, when the amino acids oxidize, we will be expecting to see the retention time shift to the earlier because of the change of the hydrophobicity. And again, we look at MS1 and look at the iso pattern and very good MS fermentation coverage. All those help us to identify the true positive. And the one last but not the least that we're talking about for the PTMS deamidation is one of the most important hotspot PTMS we will be constantly looking at, and it's also trickier. So deamidation, this is one for the asparagine deamidation. This is just the scheme for it. As you can see, asparagine will go through the loss of ammonium and become a succinimide intermediate, and then convert to either aspartic acid or isoaspartic acid. And normally they come out with a ratio of iso D and D with a 3:1, and then with the possible identification of succinimide. So this whole process is quite affected by the pH. So if you're using high pH, that's the pH 8, you will be seeing much more of the deamidated version instead of a succinimide. And you will also, of course, the high pH also introduce the artifact, deamidation artifact. So if you use lower pH, you will be able to preserve more of the succinimide version. So then you will be expecting to see more succinimide in your sample. So these are example of how they behave. So unlike the other PTMS, you will be expecting to see a certain pattern of the retention time shift. But with the deamidation, it's not so absolute. It sometimes comes like this one. It comes both the iso D and D are present and come after the wild type. And in the middle picture, you can see one of them, the iso D come before and the D come after the wild type. And then some of them also coming, like in a reverse case that the D is coming ahead of iso D. So this has a lot to do with the where the asparagine locate and the microenvironment, and if they are at N-terminal or not. So all those will need to be taken into account when you analyze the data. And of course, the MS1 and isotope pattern will definitely have to be carefully evaluated in this case. Especially deamidation is really small amount of medication. You definitely need to pay close attention to it. Another very major PTMS is glycosylation. So here we are talking about both N-glycosylation and O-glycosylation. So the workflow will be like the others, like typical peptide mapping without deglycosylation. So this is a typical HCD spectrum of the FC conserved N2 asparagine 279 position, as you can see that. Since N-glycosylation, it follows a typical conserved motif, so it's relatively easier to identify if you don't have several motifs in one peptide. If you see the oxonium ions here and then everything match up, you can be pretty sure this is your modification site, and this is the based on the mass shift and also the oxonium ion peptide map here, you will be able to identify which glycosylation pattern is in this specific spectrum. And another thing we can help with identifying the location of the glycosylation, except the motif, is so with HCD fragmentation with glycan peptides, what happens normally is there's a neutral loss of like a glycan. So you will be seeing some, as you can see here, it has this wave in front of Y5 that indicate that there's a neutral loss. But luckily for this spectrum, we still see a 200 in this specific setting. We still see a 203 of adducts, which is indicating one of the glycan residues is still attached to asparagine. So that help us with identifying the position. So with this both information ahead, we can be 100% sure this is the, of course, also this typical N297 is the location. So we are sure that this is the glycosylation, and with information, we can do a quantification with them. So this is something that you'll be expecting to see if you come getting a report from us. So it depends whether you would like this way or not, of course. So here we will be giving the sequence that identify with the glycosylation and different kind of glycosylation, and also the cartoon version of it. Then also we indicate the relative percentage of the modification. And what I've shown is in FC region, of course, we work on all over the whole region of the antibody or any other glycan proteins as well. So this is just an example of the one of the unglycosylation site on the heavy chain CDR2 region, and similar things can be provided. So N-glycan is relatively easy, and most of the time it can be done with HCD fragmentation, but O-glycosylation is a little bit a tougher case to solve. So this is one example of one peptide with two parts, because O-glycosylation happens with serine or threonine, the pronunciation threonine, the ST, these two position. So most of the times one peptide will have several of those amino acids. So if you don't have enough good fragmentation, you will not be able to correctly locate the modification site. So the first example is the HCD spectrum of one glycan modified peptides. As you can see, we have two T residue here. And if you look through the whole spectrum, you will see that everything is with the neutral loss, everything. So if you are looking at compare this spectrum together with the wild type, you will be seeing pretty much exactly the same, except the oxonium ions, because all of them would be exactly the same as without the glycan modification. So in this case, the software identified that is T10 just because the existence of the other evidence help with the identification. Because otherwise, just looking at this spectrum, you wouldn't be able to identify whether it's T10 or T5. Like depends on whatever I'm looking at. So whether it's T5 or T9 here. And then with the help of ETHCD, which the CZ fragmentation, which keep the glycan residues. So here you can see with the B1 and the CZ ions, we can see a really good C4, C5 that help us identify, okay, this T residue is not modified. And then the C9, C10, and all the following of the fragments help us to identify, okay, this T is actually the modification site. And so this is just with one glycan, and it's only one chance, right? But when the glycan is more complex, if we come across with the HCD like before, and then we have only this spectrum, and then we have like multiple possibilities, because it can be each of them has different glycan modification, and all together contribute to the final structure identified, or glycan mass shift identified here. So luckily with this HCD, what we can see is, of course, the oxonium ions help us identify this as a glycan peptide. And then here, with this specific HCD, we are seeing the Y ions of Y9 and Y11 difference. So this help us to know Y11 with the glycan residue, help us to know, okay, this T9, this position is, there is a glycosylation on this side, but it does not help us to know whether there is also glycosylation on this side or not, because everything related to this side is neutral loss. And then we look at the ETHCD spectrum that it gives us a really good CZ ion. As you can see, this C4C5 that confirm that there's no glycosylation on this side. And again, the CA9, C8 and C9 help us to identify the glycosylation on this specific residue. So this together with the last spectrum, and of course there are also existence of some other forms of glycan or glycan in the same peptides. All those together help us to identify that only this residue on these peptides is modified with different glycans. And there are also cases that so just what I want to say here is like even with SCD, ETHD, we cannot fully depend on the software to give us the correct identification or localization identification. This is just the one example that is a similar peptide as last one, but with one miscleavage. So sorry about the resolution of this spectrum, it doesn't look so great. So all the glycosylation pattern is identified. However, the site, this software gives to identify this position as the glycosylation. I want to say here T9 is not the same as last one because this one has some more miscleavage on the front. So the T9 is actually the T5 like last time, and here the second one is the T9 on last peptide. And so just on this individual spectrum, you will be looking at that you will see there's no clear fragmentation between in the whole region. The only thing that is showing up is the Y ions, that is Y10. This can be, as I mentioned, this can be Y10 or can also be Y10 with a neutral loss. So just with this individual one, you wouldn't be able to make sure that which site exactly is modified. But together with all the rest of it, we will know that for this specific antibodies, for this position, we know is the second T that is all glycosylated. So overall, for unglycosylation is relatively easier and less time consuming, but for all glycosylation, definitely need to put more input, and the manual interpretation need to be to a much greater extent and in more detail. So another analysis that I have mentioned before is part of the characterization, which is disulfide bond analysis, and I think this part of the very important character of the antibodies, the shuffled disulfide bonds or incorrect paired one will cause we will have some safety and efficacy problems. So the way we do disulfide bond analysis is different from the typical peptide mapping workflow. Here we do use trypsin or common trypsin, or sometimes a mix of trypsin, common trypsin. And the biggest difference is that we don't do reduction because we would like to maintain the disulfide bond. And then things sometimes, normally there's always some kind of free existing in a small portion of in the sample, and we will try to block it with a NAM reagent to avoid any possible disulfide, any possible shuffling that happens react with the free cysteine during the sample prep. And we will also be using a low pH and a shorter digestion time to avoid the artifacts coming from the sample prep. And on the right, it's just example of 4 different kind of IgGs, and it is a typical disulfide binding. So here, this is one example of a disulfide bond analysis with SCD. As you can see, with disulfide bond, because it's really huge, and with HCD, it will give you, with this like fragmentation, it will give you pretty good fragmentation, as long as it's not way too big, as long as the disulfide bond is not like 100 amino acids altogether, you will be able to get a pretty good fragmentation with HCD. And with this, we will be able to see a very good identification. And since it's disulfide, the only possibility is the cysteine, so there's no localization problem here. However, we do notice that it's very common that with disulfide, because of the bond existence, the fragmentation of B ion and Y ion will be focusing on, will be like coming more towards the both side of them. But this is a good enough spectrum to help us to identify the disulfide bond. And I'm not showing the MS1, the retention time thing, because when you form disulfide, there's no wild type to compare to. So we don't have to look really compare it with wild type, but we do also look at the isotope pattern to make sure it's everything is normal, not like some really abnormal patterns coming out of with the isotope. And of course, even though this is a very good identification of disulfide bond, we also have other techniques can help us to get better coverage with the disulfide bond analysis. We can use ETHCD and ETD. As you look at here, so compared to the HCD, ETD and ETHCD, it cleavage the disulfide bond, fragment disulfide bond first. So not first, like a fragment disulfide bond. So you will be able then with the HCD analysis with the fragmentation coming later, you will be able to have the peptide, two different kind of peptides. Of course, here it only show pep 2,3, but this actually all those are like the peptide signal here for two individual peptide signal here. And then you'll be able to get a better fragmentation of each of the peptides. Hence, you will get more coverage on those BY ions on different regions. So this is the ETH example and ETD. And so those when we come to have some difficult case that to analyze whether disulfide bond, these two techniques can come in really handy. And so those are like for identifying these techniques can be used to identify your expected disulfide bond and also to identify and quantify the shuffle disulfide bond, and we'll be able to give a relative quantification. One thing to note here is the quantification is really relative because from my perspective, if you look at if this is one disulfide bond, if there's a shuffle disulfide bond with this cysteine, the upper cysteine are connected to a really small like 4 amino acid, and then the ionization efficiency between that disulfide bond and this will be different. So the quantification we can do is based on the peak area that obtained from both of the different disulfide bonds, and we use that for relative quantification. But I wouldn't count on that for absolute. I would say the most useful case for this is, of course, that is one benchmark you can have for any following analysis. But if you really want to, this kind of quantification is really useful if you want to look at between different samples, stress samples or different treatments, you want to see the effect of it on the disulfide bond shuffling, then those kind of quantification will give you a really accurate a trend analysis to help you to understand the nature of your samples and solve some of the problems you might have during the process development. Yeah, so this is for disulfide. And there's another analysis that we also offer. This one last but not the least, it's one of the most complex and difficult analysis, sequence variance analysis. So with those analysis, the typical workflow will also be peptide mapping. In this case, we will try to get the most complete cleavage to avoid too much of miscleavage, because that will affect with the analysis as well following. And we would try to definitely want to use and these two enzymes to get a full sequence coverage in order to be able to identify to see all the possible sequence variants that coming on each of the position. And so this just I mark all the because it's some samples that not relevant. So you normally will see is like we'll give you the sequence coverage will be expected to be 100%. And if two enzymes not enough, we'll be looking at where exactly is the gap, what is causing it, and theoretically also add another enzyme to get 100% sequence coverage. But most of the time, chymotrypsin and trypsin both together do a pretty good job of getting 100% of sequence coverage. With this, that this is like first step, we check if the data is good enough for further analysis. So sorry, with this, we will go ahead and the data set will be submitted to TMI BIOS for the sequence variance analysis. And this is one example. So in all my previous slides, I did not show you how the MS1 rotation time this XIC look like, the XIC of each of the wild type and subunit and the isotope pattern here. So with this specific variance here, we see that, of course, when you are expecting a sequence variance to happen to your amino acid or to a peptide, you will be expecting also retention time shift because of change of the amino acid. And then, but because it's only one amino acid change, you wouldn't be expecting a major change of your isotope pattern. So we will look at the retention time shift and look at the isotope pattern. And also, of course, also look at the arrow bar, but I'm not showing here. Those are the basic things we'll definitely look at when we're analyzing each of the spectrum. And then with a good MS2 spectrum, confirm the fragmentation, have a good coverage of the whole, across the whole peptides, and then specifically at the position that the substitution is expected to happen, is not expected, it was estimated to happen here. Then with all those information at hand, we can consider this as true positive. However, there are cases that things are not so ideal. So for example, if you look at the sequence variance, which is coming with, so the previous one was also just with the HCD fragmentation. And here, if you look at the HCD only fragmentation, you will see this sequence variance was identified and labeled for threonine to serine in this position. But when you take a deeper look at the spectrum, you will see there is no clear fragmentation in this SCE region. And then with the mass shift, we are seeing with 14 Dalton difference, 14.0157. The variance that possible in this region that also matching this mass shift, we can see there are 4 different kind of sequence variants that are matching the mass shift, and 2 are actually happening in this, which is one is S, one is E. So just based on this spectrum itself, we cannot really be sure that the sequence variance is happening on this location. But luckily we have ETHCD. So we also look at the CZ ion and all the overall fragmentation together. As you can see in this specific ETHCD spectrum, we see a really good fragmentation across the region that we are really concerned about. And of course, also all the other regions. And then with those information, and then with the difference between during the position 4, 5, 6, and then we are able to identify that the only possible sequence variance happening that matching the mass shift is serine to threonine substitution. So this just one example. In actually, these are all good cases, but there are also cases that the huge amount of manual interpretation is required because sometimes there's just not good enough ETHCD fragmentations. When this happens, we sometimes either we do manual interpretation or we rerun the experiments with different settings, try to obtain a really good fragmentation to help us to identify. And because sequence variance has the possibilities is huge compared to PTMS or glycosylation, is huge. And then so this specific analysis will require a lot of human attention and the manual interpretation. So we need to pay extra attention when we actually deal with this kind of cases in order to get a confirmed identification and quantification. And so here the quantification is, of course, again, by the peak area analysis. This is the overall of the analysis that I've been talked about and the things that we are currently offering. Of course, we also, so there's one thing that I did not mention, which is peptide mapping. In a case that we give you a map and listing all the identified peptides in a sequential order. So if you ever need that, we can also do that. And what I want to mention is all those what I've shown here are typical cases, and what we are so those are just tools that can help you to answer all kinds of questions, not only to characterize your antibodies. It can be used for any other proteins, and also can also be used to, let's say, if you are not looking at the deamination oxide, if you're looking at phosphorylation, carboxylation, all those kind of PTMS, we can also do that. And if you are looking at, let's say, for some, you are doing some conjugation and you want to know the conjugation site and you want to know your labeling efficiency, all those can be used as well. So we are going to use all those techniques to try to help you to answer all the questions you have. And then if you have something that is really customer does not fall into particularly any of those, we are really happy. We are like really looking forward to hear from you, because that is something that new that help us to actually advance our technique. And we are always very interested to help customers to solve some really interesting problems they have during the discovery process. And we do have some like minimal requirement of our sample. We require minimum 20 microgram for each of the analysis, and preferably of 90% purity. But if you have something with 80% purity, we can also work with that. And the timeline, everything really depends on the complex of your sample, the complex of analysis. Yeah, so this is like overall what we do for now. And then, yeah, if you have any questions. Anthony, I think we have some Q&A.
Yes, we do.
Yes.
Yeah, there's a
Okay.
handful of questions here that I've seen come in.
Okay, so we do have some we do have some questions already, like people submitted beforehand. So we'll answer those questions first and go to the online one. So the first question people ask is, do you deal with false positive PDMS? Yes, of course we do. We do see a lot of false positive, and we have to like manually check them. So software do have some scoring or sorting the tools you can use to help to reduce the amount of work you need, but still manual interpretation is required. Yes, there are a lot of artificial PDMs can be introduced. For example, N-terminal cyclization, oxidation, and deamidation that will also be introduced during the sample prep. So as I mentioned before, care need to be taken when dealing with those kind of PDMS analysis. So the glycan profiling in different levels. So as I mentioned before, currently there are like many ways of looking at it. For example, intact level, subunit level, peptide level, and there's also release glycan analysis. They all have its pros and cons. For example, if you look at intact level, you wouldn't be able to see in such a detail with all kinds of possible, all kinds of glycans in a relative percentage, because you will only being able to see the really major one. But it's helpful if you are doing a high throughput screening, only care about the major one, and that I would suggest using Intec. And then if you are actually having many different kind of glycosylation site, including O-glycan, N-glycan, maybe many O-glycan, many N-glycan, then looking at the whole molecular weight will be way too complex. You wouldn't be able to get many meaningful information. So that I would suggest go to peptide level, which will give you the information of the site and each site, what are the distribution of the glycosylation. And then there's another method to be used with the release glycan with fluorescent detector. That will give you universal glycan analysis, but still you won't be able to get a site specific unless you know you only have one modification site. And those, unless you only one, so that helps with understanding. So those are the different, depends what exactly is your purpose of looking at the glycosylation with the different methods you can use. Yeah, 100% sequence coverage. Yes, we try to use different enzymes to try to cover it, because not every enzyme is going to get 100% by nature. Yes, so sequence variants are looking at the single amino acid substitutions, but with the technology, with the platform we have, we are able to look at the clipping, truncation, additional chains, and sequence reversal, not necessary with the characterization service, but we also have a de novo sequencing service, which can be really helpful with understanding additional chains, because if you do have a sequence of additional chains, we can help you to answer it with the characterization. But if you don't, and if you're suspecting some sequence reversal or some sequence not matching, we can use de novo sequencing, that is a remap service, to help you to understand to solve your problems, and together with the clipping and truncation. Yes, we can. Really depends on it's really case by case. Apart from the intact that I've mentioned before, there's another technique you can use is disulfide. If you are suspecting, for example, if your one light chain is kappa, the other light chain is lambda, then you will be suspecting a different disulfide bond that will help you to understand the mispairing as well. Turnaround time, as I've mentioned, maybe Anthony, you have a better idea to this question.
Yeah, I mean, at this time, it's generally a couple of weeks for each individual analysis. If you want to do like quite a few different analyses, it take a couple more weeks. Pardon me, I'm trying to fight off a sneeze. And generally we have no lead time at the moment either, so you don't have to worry about like months or weeks of advance notice. Like we can take your samples today, we can process them the next day, and we can start that analysis, and it'll take generally a couple of weeks to do that.
Cool.
And I think this one's for me too. In the greater context, how are the insights from the LC-MS antibody characterization or protein characterization workflows like critical towards like early stage R&D and regulatory compliance? I think I addressed some of these earlier, but I mean, I can certainly say it again. There are just like some ways in which understanding the functionally expressed molecule, the true protein that you're trying to work with, ways of understanding it through the LC-MS based characterization that can influence the ultimate engineering and final form of the molecule of interest, right? I think Jinrui has given a lot of really good examples with respect to intact mass analysis, ensuring that whatever is producing the protein of interest is truly just producing the protein of interest and not some other variant as well. And PTM analysis, I think, is really critical in that as well, and ensuring that that the finally expressed protein is the same today as it is 6 months from now, a year from now, in such ways, and certainly with respect to like regulatory compliance, doing that work early on, showing that this due diligence has been a part of the overall antibody or protein development life cycle since inception, certainly engenders some confidence and trust in your team and certainly in the molecule itself long term as well. So I think that we can always have a more detailed discussion afterwards as well if needed.
Cool. I see some question coming for the online version. Can you calculate out percentage of each species with different molecular weight based on the peak observed on? Yes, if the species is observed on the intact level, we will be able to calculate the percentage of them. For example, for glycan, you will be able to see the percentage of G0F and G1F and the combination of them. The second question is, what is the resolution of a message tag? So we use Orbitrap. Like we use the really advanced the thermal tribrid version that you can define your resolution. So for us, we normally use 60. Let me make sure I'm not getting the missing 10. I would say 60,000 for the MS1, and we can use 15,000 or even 30,000, even 50, depends. Of course, the higher resolution means the longer time, but with 15,000, you will be able to get pretty much everything you need for this analysis. Regarding mispairing analysis, is it possible to evaluate mispairing between endogenous trends and transgenic trends in case of engineering? Ooh, this is a question that I think if you are interested, we should talk like more into because
I think we need a bit more detail on this one as well.
Yes, yeah, definitely.
If it's a need a cell line, it's a different question, but it sounds like it might not just be like a cell line. It might be like an endogenously expressed protein of interest, and like it's kind of a gene factor approach. So yeah.
Feel free to just send us an email, then we can discuss a bit further. So any advantage of doing glycosylation analysis by HCD over ETHCD? So it's some lab does not have ETHCD, so you can only reach HCD. And then if you're doing N-glycan, I wouldn't be too concerned about the use of ETHCD because quite often you will get pretty good fermentation with HCD. But yeah, there is also a possibility of the localization problems that you will be coming across even with N-glycan, that HCD will help. But HCD is faster. each of the HC scan is like each of the ETHC scan takes much more time than HCD. So if you're actually having a limitation of how much time you can spend for the MS2 for the glycosylation, you might want to go with HCD. But generally, our like default setup is we'll be looking at ETS and HCD and ETHCD for both. So okay, so to convert, can you go back, take a look at the spectrum? So there's was a question about go back and take a look at the spectrum.
I think it's from a sequence variance analysis.
Comment more on the time spent on the time spent. So for sequence variance, it's I would say a lot of time will have to spend on manual interpretation because there's so many possibilities. And then the software really do a very good job to help you already to give you the certain possible possibilities. It's one of the best, but you still need to spend a lot of time to manually interpret. And especially one thing, if you do see that the software gives you a match of one specific sequence variance, but you see one major peak or a couple of major peaks that is coming out of your spectra that are not assigned, and you need to be alert because that most likely indicates there is a modification happen to this spectrum, but not necessarily the localization of the mutation is correctly labeled, correctly assigned. So yeah, so with the sequence variance, the one that really I can't really say about the time exactly because it really case by case, but I would say generally a lot. So that's pretty much all the questions I have.
Yep, that's all of them.
So that's a lot of questions, and conveniently, we're also at time. So thank you everyone for joining us today, and thank you for submitting those awesome questions. This webinar was recorded, so you will be receiving the recording so that you could rewatch at a later time. And then if you have any further questions, feel free to email us offline. But yeah, have a great day, everybody.
Great. Thanks much. Bye-bye.
Thank you. Bye-bye.